copy it. There you go. And okay. I got your email. I just haven't gone around to replying to it yet.
No worries. Thank you.
Yeah, sounds a lot more fun there than it is here.
I'm in a local coffee shop where I live, and it's super cute.
Oh, neat. That's
Yeah,
nice. That's a good way to study.
it's awesome. I'll show you real quick.
Wow.
And the people here are so nice, and it's like it's a small town, so we know everybody.
Cool. And you can get coffee whenever you want too.
They just bring it to your table. It's really nice.
Well, this is my surroundings. Oh, actually, you can see my feet. Oh, that's a little bit embarrassing. I forgot about that. Probably
Yeah,
don't
but
want to spend too much on that.
you've got your doggies though.
Yeah, I like rub her back with my foot while I'm teaching.
That's the life.
Okay, so where are we at? We are doop doop doo. We're just finishing up RNA processing. Dum dum dum. Scroll, scroll, scroll. Okay, so this is kind of like a summary picture in a sense, right? What we've gone through so far has been capping the 5' MPG or the methylguanosine cap, right, which you can see here. Gone through splicing, which is a fairly involved process, which also has to be very accurate. And the very last thing to do is to add this poly A tail, right? And so poly A tail is essentially just a series of A's, right? And the reason for having this poly A tail is that, again, the 3' end of the mRNA is vulnerable to degradation, right? And so if you want your mRNA to hang around so you can translate it into protein, which is usually the purpose of producing it, then you have to protect this end somehow. And this is kind of like a buffer, like a telomere, but it's not really as well, because this isn't long so they can get chewed up before the mRNA gets degraded. This is long because it provides a binding site for what are called poly A binding proteins. Let's get the chat up. I can write that down. Ah, soon you're a pain in the ass. Oops. So those proteins bind to the poly A tail and protect it from RNases. And as I was talking about a little bit at the end of last class, modulating that is actually a way in which you can control the abundance of an mRNA transcript. So we can actually, in part of our control of gene expression, I want to kind of keep giving you like these like teasers, I guess, as we go through. If you can eject those poly A binding proteins, you can actually get that transcript degraded so that essentially you shut down gene expression, right? And so this is a dynamic process. It's not something which is static. The poly A tail obviously is, right? That's just there because it's put there. But how that is used kind of varies depending on the transcript and the context in which that transcript exists. And so essentially that builds off of the cleavage. So if you remember about RNA transcription termination, do we have that in here? No, that's in a different PowerPoint. Too lazy to go open that up. Where the pre-mRNA is cleaved by an endonuclease, and then RAT1 does its job and kicks the RNA polymerase off. Well, that cleavage site, which you can see just here, essentially that's downstream of a consensus sequence. Again, this is a recognition site for the enzyme, for the RNAs. When that is cleaved, that 3' end serves as the starting point for the adding of these series of A's, this poly A tail. And actually, I think from what I've been reading, and it's quite confusing to be honest, there's an awful lot of stuff going on. But the proteins that cleave this also serve to start producing the poly A tail. I don't actually truly understand how that works because I haven't done enough reading on it. But this is all part of like the same process essentially. Just so showing you it in sequence because that kind of makes it a little clearer to see what's going on. And essentially once that's finished, and this is obviously the very last thing to be done because that's at the end of transcription when transcription is being terminated, then this protein is now available to be exported from the nucleus to the cytoplasm. That's the very, very last step. And that's actually quite a lot more complex than I'm really willing to go into right now. It uses proteins that are kind of associated with the splice junctions, right? These junctions between the exons. And essentially they find their way to the nuclear pore in the membrane and then get threaded through and into the other side. But there's a lot going on there, which I think would just muddy the waters. All you really need to know is that it's pushed out into the cytoplasm and therefore it'll be available for ribosomes to assemble on it. And that's pretty much it for RNA, right? So essentially this is what you end up with at the end of transcription. And this again is specific to eukaryotes, right? Prokaryotes do things very differently in many ways, which we'll see next section. Does anybody have any questions about transcription or RNA processing? We most certainly, I think we've got an exam next Thursday. Yeah, we've got an exam next Thursday. So if we make good progress on translation today, which we should do, it's a lot to cover, but it's kind of a straight shot essentially, then part of Tuesday will be available to go over any questions you have about the material. So I'd strongly, strongly recommend, if you haven't started doing so already, from now on, essentially, you know, over the next, 6 days, 4 days, if we're talking about lecture on Tuesday, is go through all the material and just pick out all the stuff that doesn't quite make sense, right? So you have a list of stuff to bring with you and you can keep me loaded with questions, right? So to get the most out of that session. Okay. All right, so when we get to translation, really translation is the whole process of, where'd that picture go? Here we go. Taking this mRNA, assembling a ribosome on it, right? And that's kind of a sequential process. It differs again between prokaryotes and eukaryotes a little bit. And then that ribosome will process along this mRNA transcript until it gets to, well, again, this differs from prokaryotes and eukaryotes. Anyway, at some point, we're going to hit the start codon, and we're going to start translating those words of information into an amino acid sequence in a polypeptide chain. That always starts at the start codon. That's why it's called start codon. Kind of self-explanatory, right? And that is always an AUG, which corresponds to methionine. Where's my chat box gone? Get that back up here. Oh, Tanya, you are there. Oh, there you are. Yeah, chat. So every single protein always starts with a methionine. Doesn't necessarily have to stay there, right? You could cut it off at a later point, but in the process of translation, the very first amino acid to be added is always a methionine, right? And so if you wish to try and figure out where a gene is in a stretch of DNA, this is what you look for, right? And then you see, oh, do we have some introns? And at some point, we're going to find a start codon in that frame, right? And so a lot of the words we're going to be using here are kind of very specific to this context, and they're very useful to know, right? So codon, I'm almost certain you already know what that is, but just in case you need a refresher, just rearrange them. Get off it. Sorry, trying to rearrange my, there we go. So a codon is a 3-letter sequence of nucleotides or bases, right? And so essentially what that is a word, right? And so if we use the analogy of letters, words, writing, et cetera, right? The RNA bases in an mRNA are the letters, the codons are the words, and then when you string enough of them together, you get like a paragraph, right? That's what your protein will become. And so that gives you a frame, right? Essentially the odor of the words equals the frame, or really better term is the reading frame. So if you look in this picture here, if you remember, I gave you a definition of what ORF means. It's the open reading frame. And so that means that once you start that with an AUG, you then read it in 3-letter chunks. So the first AUG is the first amino acid. The second 3-letter word is a second amino acid. The third 3-letter word is a third amino acid, and so on, right? That gives you your frame, right? And because we have double-stranded RNA and you can have 3-letter words, technically there's 3 possible frames going one way and 3 possible frames going the other way, right? This is why it's important to specify which one you're using, and that's specified by this start codon. Also, if you look far enough down the mRNA, you will see in the same frame, right? So this is in the 3, 3, 3, 3, 3, 3, 3, all the way down, right? The last 3 will be a UAA, a UAG, or UGA. These are the stop codons. These do not code for amino acids. And so they pause translation until something comes in to kick the ribosome off. Because there's no tRNA to come in there and add an amino acid, right? So translation stops at these codons. Okay, so this is a general overall context, but there's an awful lot of different, bits and pieces and nuts and bolts in this. And really we have, yeah, here we go, a bunch of different major components. Obviously we have the mRNA, that's the information. That's kind of a given. We also have things called transfer RNAs, shortened to tRNAs. Has anybody heard of the Rosetta Stone, just out of curiosity? Right, so it's the tablets, I think, probably found in the Middle East somewhere, Eastern Mediterranean maybe, presumably in a place called Rosetta. Essentially allowed hieroglyphics to be translated into, I think, Aramaic or Sumerian, I can't remember which. But they were basically the means by which you could take one form of information, like pictorial, which were hieroglyphics, and turn that into a different one, which is somewhat something that we could relate to. And so essentially, tRNAs are that, right? They take the 3-letter words of the mRNA and translate that into one very specific amino acid. And we'll get to how they do that later, but it's essentially based on part of their sequence. So part of the tRNA is directly, well, 3 bases is the direct complement of the codon that they recognize. So if you have a codon, almost wrote Canadian in shorthand there. You have a codon in the mRNA and have the anti-codon in tRNA. And so, for example, UAG, right? AUG, sorry, there. In mRNA, oh, come on, would be, what would that be? UAC in tRNA. And again, kind of getting back to that kind of principle that we've been talking about a whole bunch all the way through, that of base pair complementarity, right? A's bind to U's or T's, C's and G's, right? It's important in DNA replication, it's important in transcription, and it's also important in translation because we're going to be using that to very precisely bind a tRNA to that mRNA. So really everybody thinks about ribosomes being the sexy sauce when it comes to translation. Actually, it's tRNAs that are the most important part. You know, if one were to champion one particular part of translation, which I guess you'd have to be a serious nerd to do that, but anyway. And so the piece which well, essentially, I don't know, enforces that specificity are these enzymes here called amino tRNA synthetases. What these do is they recognize that anticodon on a tRNA, and they very specifically add the amino acid that corresponds to that anticodon to the end of the tRNA. So only one tRNA recognizes one codon and carries one amino acid. You cannot have one tRNA being loaded with more than one amino acid. Otherwise, this whole thing breaks down, right? So it's super, super, super specific, right? The anticodon is both determines what codon it binds to in the mRNA, but also what amino acid gets added to it by the amino acyl tRNA synthetases. There. Hopefully I won't have to talk about these very much because they're a really long word. Okay, so this is also part of that specificity. And then you've got ribosomes, and ribosomes are just a bunch of proteins and RNAs that do stuff. Essentially, their job is simply to form a peptide bond between the incoming amino acid and the existing polypeptide chain, and then cleave that amino acid from the tRNA. So that's their job, right? And we'll get to how they do that in a second. I'm getting jealous of Kim. I really want a cup of coffee. I've got the coffee maker already loaded up, but it's over there. I should actually have it right next to my little office space. That would be a far better idea. Okay, so a few more words, right? Polypeptide chains. Essentially, what would the best way of putting this be? So the polypeptide is a linear sequence of amino acids. Protein, equals a 3D shape that has a function. Right, and so that's an important distinction to make because the polypeptide is really the string of amino acids that is synthesized by the ribosome. That is not the final part of translation because that protein has to fold into the correct conformation. And so those vary hugely. Oh no, go away. There you go, that's what I wanted. So for example, we have myosin, which is one of the two parts that makes up your muscle fibers, which is actomyosin. And myosin has this really funky shape, right? Where essentially it's a whole series of long, twisted polypeptide chains. And then you have this head at the end. And this is the part that actually does the movement, which allows the muscle fibers to slide over each other. And essentially your muscles shorten. And that's how you contract your muscles, which is cool. I love talking about muscles. They're super fun things to talk about. Actin, on the other hand, at the top has this kind of long, slow spiral. So actin is made up of two bits of an actin protein, right, which are spiraled over each other. That essentially is a ladder along which the myosin walks. And myosin basically does that. And that's how it translates basically ATP energy into mechanical movement, right? Because it changes its conformation. So that's one type of three-dimensional structure, right? It's very long, kind of globular end to it. You've got, what can we have a look at? Hemoglobin, spelled in the proper way, as you can tell. None of this taking letters out of words that you Americans do. And so hemoglobin is all kinds of funky, right? So this is actually what's called a quaternary structure, which means that it has, these are individual proteins, right? So hemoglobin, a thing that carries oxygen around your blood, consists of two pairs of two proteins, alpha chain and beta chain. So there's 4 in total. Each of these has at its center what's called a heme group, which is there we go. This is what a heme group looks like. It's basically a funky ass cyclic what's it, right? I think that's the precise term, which has an iron in the middle of it, right? And so by changing the shape ever so slightly, if you actually look at 3D models of hemoglobin, you see that it flexes, oxygen bound, oxygen not bound, oxygen bound, oxygen not bound, right? And so its actual shape changes. And this is what it looks like if you look at a kind of a ribbon model. Uh-oh, where's that taking me? Oh, Australian government website. Well, that's weird. Anyway, so that's what it looks like in a kind of bubble model. And so the point that I really want to get across, where's that picture? There it is. No, that's not blow upable. Well, that's disappointing. But anyway, the point I want to get is that proteins have a very, very, very large array of shapes and sizes. Oh, here's another cool one to look at. This is what ATP synthase looks like, right? And so this is probably one of the more complex proteins you'll ever see, in that it has this is probably an alpha helix barrel. And so it's a ring of kind of helix. If you think about like a series of piggy tails kind of chopped off and arranged into a ring like that, that sits in the membrane, actually the inner membrane of your mitochondria. There's another protein which goes through that, and then there's a bunch of different proteins come together. I can't remember, there's probably about 30 proteins involved in one ATP synthase. And there's another one which actually holds this whole thing together, right? And this part actually physically rotates, right? It's quite astonishing, right? And this rotational movement is what generates ATP. It's actually a mechanical rotation, which I think is extraordinary, right? And so there's a huge array of different shapes and forms of proteins. They all have very different properties, and those properties are defined by the amino acid sequence, right? So that polypeptide sequence, which is encoded by the mRNA, contains all that information. But that long chain of amino acids is not the final product, because that has to kind of basically fold and twist and turn into the final thing, which could look something as complicated as this, right? And so the properties of those amino acids also give those proteins their properties too, right? And so this is a great example, actually, just by chance. This is a phospholipid bilayer. This is a membrane here in gray. One love. Oh, that's bright. And as you probably know from kind of basic cell biology, Gen Bio 1, the inside of membranes is very hydrophobic, right? That's actually why this whole thing works, because protons can't get across this on their own. And so you would expect the amino acids in this particular region of that protein to also be hydrophobic, right? Because otherwise it wouldn't work, right? It wouldn't be able to sit there. And so, I'm kind of bouncing around a little bit. We've got I'm going to blow this up a touch because this is teeny tiny. My eyes can't read it. I need new glasses. And so amino acids, and there are 20 of them. I always think there's 21 for some reason. Same as I always think that there's more states in the US than there are states. I don't know why. Blame that on a Will Smith film, personally. Anyway, I don't know if any of you have watched that 51st state. How many states are there? Is it 50 or 51 in the US? Oh,
50.
50. Kim's like, oh shit, I don't know. 50, yeah. So the film's called 51st State. It's a really good film, actually. I like Will Smith. Anyway, so let's bounce back a little bit because this is going to be useful. So this is essentially what an amino acid looks like in basic form, right? And it's called an amino acid because there's an amine group or amino group, which is a nitrogen with a number of hydrogens attached to it. There's a acid group, a carboxyl group at the other end, and those are joined together with a carbon called the alpha carbon. This constitutes the backbone of the amino acid and what will actually make the backbone of the protein, right? The actual chain part. And this is very much analogous to the phosphate sugar backbone in DNA, right? This is the same for all amino acids, just as the phosphate sugar backbone is the same for all bases, right? And so this is always the same. It's sorry, distracted by kids. The R group, the side chain, is what differs. And so this is what gives amino acids their individual properties. And those properties are extraordinarily important, right? Those are really what define how the protein folds and kind of what it does, right? So this stays the same. Oh, actually, another kind of, all right, a little bit scatterbrain this morning. This is also the basis of the directionality of amino acids. Actually, where the hell did that picture go? No, I think that was in another PowerPoint. So DNA has directionality, right? 5' to 3'. I mean, it's double-stranded, so it's anti-parallel, but for the purpose of a gene, which is only encoded by one strand, it's 5' to 3'. The mRNA produced by that has the same direction, right? 5' to 3'. And this is in part essentially created by the fact you can only add a new nucleotides to the 3' end, right? So you can only read it in kind of one direction. And proteins and amino acids have the same directionality, but now it's because of this OH group here, part of the carboxyl group, this is the only place where you can add new amino acids. So polypeptide chains or proteins are also synthesized in only one direction. And that's actually called the N-terminus, right? So the and the N comes from the amino, and the end is the C, and that's the carboxyl terminus. Oh man, my spelling is terrible. And so proteins also have directionality as well, both from their structure and also from the sequence in which, or the direction in which they're synthesized, which kind of makes sense, right? You know, you're going to be reading the mRNA. So you read DNA in one direction to make mRNA. You're going to be reading mRNA in one direction to make protein. So it makes sense that protein too has a particular direction. I'm going to get back to the side chains in a second. I've just realized I should tell you about this stuff first. So let's get this a wee bit smaller. So again, you can see here, right, once we've called the amino end, right, so the proper name for that is the N-terminus, the carboxyl end, the C-terminus. And essentially, it's a series of stepwise reactions. Again, the same dehydration condensation reaction. Hey, Peep, could you keep it a little quieter, please? Thanks, love. So it's a series of those sequential dehydration condensation reactions where you take the OH from the carboxyl and one of the H's from the amino group, create water, and the result is a peptide bond, right? And so this is directly analogous to the phosphodiester bond between phosphates and sugars in DNA or RNA, right? Same kind of deal. And then you do that enough times, you end up with a polypeptide, right? And depending on the sequence of the mRNA, that determines what order of what amino acids you have in that polypeptide, which in turn determines what that protein will look like and what it will do, where it will be as well. And so kind of circling back to kind of almost where I started, these R groups are one of the key determinants of what that protein is going to be like. And they're also actually one of the main reasons why mutations vary in how serious they are. Even when they change amino acids, they can vary from being a meh to being a, oh shit, that doesn't work anymore kind of mutation, right? Just depending on what gets changed to what. And so the reason for that is they have very specific properties, right? So those R groups, like the side chain, always have a fun pirate joke. What's a pirate's favorite letter? Come on. Pirate's favorite letter? Come on. No? Someone's got to say it. Tanya, go on. Can't shake your head forever. Oh, it's the first time no one's ever let me tell that joke. That's disappointing. Anyway.
Ali said R.
No, it's the C. Get it? Thank you, Kim. I appreciate that. Being the sacrificial lamb for my terrible joke.
R was better, I think.
Yeah.
Yeah, R kind of makes more sense.
But it's a pun on the letter and the word, right? Because they're pirates, they're at sea. Sea's a letter. Anyway. Moving swiftly on. So large class is hydrophobic, right? And so these have essentially only carbons and hydrogens, and sometimes the odd sulfur, and sometimes funky carbon rings as well, like phenylalanine. But they all have an inability to form hydrogen bonds with water or anything else for that matter, right? And so these will, well, they're hydrophobic, right? They actively try and avoid water. Where, question to the class, where would you expect to find a lot of these residues in a three-dimensional protein, let's say a globular protein, if that protein, our cellular environment is largely aqueous? Where would you expect to find the majority of hydrophobic residues in a protein?
The
As a result?
center.
Yeah, in the center, right? And they also do a very important job because you don't typically want protein water, sorry, getting into proteins because it will physically disrupt them. And so like the hydrophobic core of a protein actually helps to keep water out of proteins so that their three-dimensional structure is not changed by the presence of water molecules. And actually, if we were to throw one of you into the sea and keep you alive so you didn't drown, and send you down to, I don't know, probably even just like 500 meters, 1,500 feet, other than being a lot smaller than you originally were, because you'd be crushed by the pressure. The other issue is that pressure also forces water molecules into proteins. So even if you were an organism that could survive, like you didn't have lungs and stuff, at that pressure, you would have the issue of like the barometric pressure forcing water into your proteins to disrupt them, which is kind of cool. I'm a big fan of extremophiles, which is why I often talk about them in class. Right. So as you can see, if you have a mutation that changes a amino acid from a glycine to an alanine, it's kind of a bit of a non-event, right? They're both hydrophobic, they're both similar in size, it's not much of an issue. If it changes it from glycine to tryptophan, and I don't have a codon table to hand to see if that's likely or not, but if it did, that's a much bigger issue because of the difference in size, right? Similarly, if you went from hydrophobic to hydrophilic, right? And so these all have either OHs or NHs, right? So amine groups or hydroxyl groups, these are able to form hydrogen bonds. They'll typically be found either on the surfaces of proteins, or let's say you have a pore, right? A channel, like the one in ATP synthase, right? Protons have to go through that. That has to be a hydrophilic core. So even though it sits in a hydrophobic environment, right? The center of a membrane, on one side it's got to be hydrophobic, on the other side, on the inside, it has to be hydrophilic, otherwise those protons won't go through, right? So if you change from hydrophobic to hydrophilic or vice versa, that is a very disruptive mutation and almost certainly result in the protein not working, right? And so there's an awful lot to how these simple amino acids and these simple structures translate into like the big final product and how it works. It's astonishingly complex. And I think in fact, it's so complex, figuring out how you go from a polypeptide chain of these things to a protein that is only just very recently they've been able to figure out how to predict what protein you'll get, like the actual shape of a protein from its polypeptide sequence. And I think they used artificial intelligence or something like that to be able to figure out how to do so. Astonishingly complex, right? It just is not very simple to work out, and partly because all of these things interact with each other in ways which are very hard to predict, right? Very, well, not hard to predict, but so complex it's difficult to. And you also have, pardon me, R groups that are either negatively or positively charged, right? And these have these are actually often involved quite a lot in enzyme active sites. So you'll pretty frequently find one or more of these residues involved in, let's say, if an enzyme has a kind of a region, right, in which it's going to bring a substrate in, the amino acids that line that region, right, that surface, I guess, you're very likely to find positively or negatively charged amino acids there because they're able to interact with the substrate, hold it in place, and do stuff. That's kind of getting into the realm of biochemistry, which is not my strong suit. And finally, the weird one of all of them is cysteine. Actually, I like talking about cysteines because I was going to say something sad about my life not being full of excitement and stuff, but actually a gene or a protein that I worked on for a good chunk of my career, part of my PhD, which is involved in signaling in worms, has a bunch of cysteines, conserved cysteines in it, which hold together 2 units of that protein, right? It's a dipeptide. Or not dipeptide, yeah. 2 subunit protein. Anyway, so the cool thing about these is that these form covalent bonds with other cysteines. And that's called a disulfide bond. Oh, crikey, you got an exam after this? I'm sorry, Tanya. Just saw your chat that kind of disappears while I'm gesturing at my PowerPoint. Yeah, so good luck in your exam. Okay, that terrifies me. So anyway, cysteines form disulfide bridges. And so that's basically covalent bonds between the sulfur atoms. And they look like this. S, S, like that. And so these are cool because they are very rigid and they are very strong, right? They are not hydrogen bonds, which are weak, or even ionic bonds between basic and acidic amino acid residues. They are covalent bonds, right? And so you find those when you really need to hold stuff together, right? And so one of the, again, kind of going back to extremophiles, actually, I mean, we talked about TAC DNA polymerase in lab, right? From Thermus aquaticus, grows at 72 to 75 degrees Celsius. Essentially, at that temperature, there's a lot of energy trying to kind of basically vibrate, these proteins apart so they fall to pieces. Essentially, it's the same thing that happens when you cook an egg, right? That's why when you crack an egg into a pan, the egg white is translucent. And then as you add heat, that egg white turns white, right? And the reason is that you're denaturing the proteins, that albumin, which essentially kind of falls apart. And then when it sticks back together, it's in the wrong shape. And so it blocks light. That's why eggs turn white when you fry them or cook them, which is kind of neat. But also, if you think about it from a kind of evolutionary context, right? If you're living in a very, very hot environment, there's a lot of energy trying to denature your proteins. And so you need to hold them together more rigidly so that they retain their shape. Because proteins are supposed to move, right? But they have a limited range of movement. If they move more than that, they're broken, right? They don't work. And so you find a lot more cysteine residues in organisms from very high temperatures, because these literally hold their proteins together and stop them falling to pieces. And so the reason why I like talking about this stuff is because this is all like abstract, right? It's kind of hard to figure out how this relates to any kind of biology that you can see and touch, right? But so that's why I try and bring this into kind of bigger biology, right? And you can see how if you are existing in an extreme environment, right, there'll be a very strong selective pressure by your environment that will select for mutations that generate cysteine residues, right? Because those cysteine residues will, through the formation of those disulfide bridges, hold your proteins together, so you're more likely to survive, reproduce, pass those mutations on to your offspring. So it's kind of a neat way of thinking about how this ties into, bigger picture stuff. And this is just a kind of a cartoon picture of what a general protein could look like, right? So we've got our N-terminus and our C-terminus. These can actually be right next to each other in the finished protein. There's no, just because they happen to be at one end and the other end of a polypeptide chain has absolutely no relevance to where those ends end up in the actual protein. And these things that we're looking at here, these are actually called secondary structures. Pardon me. And these secondary structures are kind of like sub-assemblies, essentially, of the protein. So if you think of the polypeptide sequence as being the primary structure or the very first one which is produced, right, it's very much one-dimensional, right? These secondary structures are kind of small assemblies that form, right? And you've got things called, these are alpha helices or a spiral, basically. And they actually do look like this. These ones don't look like arrows. We just draw them as arrows because it kind of makes it easier to visualize them. Right? It'd be really weird if in your proteins you had a whole bunch of arrows pointing all over the place. So, but essentially what these are saying is that beta pleated sheets are flat, right? So you can have lots of, helices. You can actually make a barrel out of them, right? That's why a lot of membrane pores. Kim, order more coffee. Still got half an hour to go. The beta pleated sheets tend to be more two-dimensional, right? They tend to be kind of in more one plane, right? So they're kind of flexible like that, but they're not very flexible kind of like that, essentially, right? And so these give, in many ways, proteins their properties. And also these are all joined together with kind of non-complex strings of loops. And so these allow the protein to kind of form the correct conformation. And these are also what allow the protein to move, right? So not all, but many proteins need to move slightly in order for them to function. Hemoglobin is a classic example, right? It has to flex. Myosin is another one, right? It has to be able to physically move, right? Kind of its head has to be able to bend backwards and forwards. And so those loops allow that to happen. Okay, so those are all like the basic kind of background of proteins, like what makes a protein, how do proteins differ, and that depends on what they do and where they're at, essentially, right? Very much so. And that is in turn really very much determined by the amino acid sequence, right? What amino acids does it have? And then what shapes and form will those take when they fold together to form the final three-dimensional structure? So what I need to do now is actually go through, okay, well, how do you make those proteins? And so essentially what I need to do is kind of give you a little more background into the individual parts, and then we'll get into the mechanism. All right. Feel free to stop me at any time if you have any questions. Okay, so first of all, what is a ribosome? Okay, so a ribosome is again another large protein assembly. We've got the large subunit because it's the largest one, not surprisingly, right? This is really the site of peptide bond formation. This is where the tRNAs are going to be. And we have a small subunit. This is what binds to the mRNA. So the small subunit is fairly straightforward, right? It binds to the mRNA and it ratchets along it 3 bases at a time. The large subunit is much more complex. So this consists of both protein and ribosomal RNA. So remember, ribosomal RNA is what is responsible for the catalytic properties of a ribosome, right? The formation of the peptide bond and the severing of the amino acid from the tRNA. Now, essentially, in this large subunit, we have 3 main sites. We have the entry site, which is called the amino acyl tRNA binding site. Could also remember that as being the access site, the A site. So written in order, it's APE, from the order in which the tRNA goes in and then leaves. Then we have the P site, which is stands for the peptidyl tRNA binding site. And so I always remember that as being the peptide bond site, right? So that's essentially the peptide bond is created between amino acids in the A and the P site. Then as soon as that amino acid is kind of bonded to and then cleaved from the tRNA, the whole thing moves along, and that tRNA that does not have an amino acid anymore, right, it's discharged its amino acid, is now in the exit site or the E site, and it leaves. So there is a continual flow through of tRNAs coming in to the A site. Then essentially the whole ribosome moves along. That tRNA is now in the P site. Another tRNA comes into the A site, and it comes in based on its base pairing with the mRNA being held in a small subunit. This whole thing moves along, another codon, another tRNA comes in, tRNA leaves, comes in, leaves, right? So this constant flow through of tRNAs coming into and then leaving the ribosome and dropping off their amino acids in the process. So all of that jazz happens really in the large subunit. Okay, we'll get to in a little bit more detail that in a bit. So again, remember when I talked about RNA secondary structures? Well, tRNA is a classic example of that. And so tRNAs form, it's called a cloverleaf shape. It actually looks more like this, right? It's a little bit more complex. I always think of it looking like a bit of like a Santa hat, to be honest. And the amino acid is like the bobble on top. But essentially it consists of the main regions that you need to care about are the very top, where an amino acid is attached. Pardon me. And the very bottom, where an anticodon is present. And so again, this is a series of 3 bases that very specifically will base pair with a codon in mRNA that it matches to. So this anticodon and the amino acid in which it carries are how that information in the mRNA is translated into an amino acid sequence in a protein. Because this is very specific. You'll for any one anticodon or tRNA, you'll only have one amino acid, which means for any one codon in the mRNA, you'll only ever get one amino acid. Now, some of you already probably know, you can get the same amino acid added from having multiple different codons in the same spot. That's different. That's called degeneracy or wobble. But this is more about the specificity of one codon equals one amino acid. Mostly. We'll get to some exceptions to that later. And so the means by which that anticodon is always paired only with one amino acid is through the action of this enzyme here, right? So this is amino acyl tRNA synthetase, there, which is a very long word. And so essentially what it does is it binds a very specific amino acid. And again, it will do so based on its own shape, right? So this again is a protein, and this is essentially what it really looks like according to the current computer model, right? Has this kind of shape. But in cartoon terms, it has a very specific shape around that amino acid binding area, which will mean that it'll only bind one amino acid. So essentially we have amino acid tRNA synthetases for all 20 amino acids. There's one specific for each one. How did that get there, I wonder? And so essentially ATP is used to facilitate the binding of that amino acid. Right, so once the amino acid is bound, now the enzyme is able to bind tRNA, which is kind of shown in this sort of funky format here. And this tRNA will only be able to bind to this enzyme if it has the correct anticodon. And so essentially there's specificity at this end where the amino acid binds, and this end where the tRNA binds or the anticodon binds. And then once the tRNA comes in, then we form a bond, covalent bond between the amino acid and the tRNA, and the tRNA then leaves. It's now called a charged tRNA. And as you can imagine, when it gets to the ribosome and goes through it, it will become discharged, right? So essentially this is one large cycle, right? These tRNAs are constantly being loaded with their amino acid. They carry that amino acid to a ribosome when their codon pops up. They drop it off, that amino acid gets added to the growing polypeptide chain. It's discharged, comes back, gets recharged, and it basically goes through this continual cycle. Anybody got any questions? Questions? Okay. So as with transcription, really there's three phases: initiation, elongation, and termination. So initiation is really the basis of how does a ribosome find an mRNA and start the process, right? And this differs between prokaryotes and eukaryotes. And so one of the main differences is in prokaryotes, the ribosome assembles directly on the start codon. Whereas in eukaryotes, the ribosome assembles at the 5' cap and then processes along until it gets to the start codon. Right, so that's kind of the main difference between them. Also, the subunit sizes are a little bit different, but that's not super important to know. And so essentially what happens is we have in a prokaryotic mRNA, we have something called the Shine-Dalgarno sequence. This is a sequence in the mRNA that's just upstream of the start codon, which you can see here. And this is recognized by an initiation factor called IF3. Yeah. And essentially what that does is it brings along with it the small subunit of the ribosome, right? So remember, ribosomes consist of a large subunit, which is where the AP and E sites are, and a small subunit which binds to the mRNA. Oh, and also these want to self-assemble very easily. And so the initiation factor also prevents the assembly of the large subunit too, which is what allows the small subunit to bind with the help of IF3 to that Shine-Dalgarno sequence. Then once it does, a whole bunch of stuff comes in. One of the most important ones of which is the tRNA for methionine. And so essentially in prokaryotes, the whole complex kind of assembles on the start codon around the tRNA for methionine, which is the first amino acid. And so we get our tRNA with methionine attached to it. Again, as I pointed out earlier, this has an anticodon of UAC. And so we get direct base pairing between the start codon and the anticodon of that tRNA. We have a bunch of other stuff come in, including GTP. And so GTP, is that guanosine triphosphate, is kind of a sister version of ATP. Right, so this is another form of energy which can be used by the cell. Works in the same kind of process, right? It's the endmost phosphate bond which carries the energy, same as for ATP. And so the start tRNA essentially binds. We've already got our small subunit bound. We have another initiation factor with some energy in the form of GTP binding, right? And all of that then leaves to allow the large subunit to assemble over the tRNA. And so this tRNA is going to be in the P site, because essentially this next codon here, whatever that encodes, now we're going to have a tRNA come in with an amino acid corresponding to that codon. Then we're going to have a peptide bond formed between methionine and that amino acid. And then the ribosome shifts along one codon, a new tRNA comes in, peptide bond forms, and now the original tRNA, the one from methionine, leaves. Right, and so for prokaryotes, it's fairly straightforward. Anybody have questions? Yep. Cool. Okay. And so for elongation, it's actually worth looking up like videos and stuff. There's one link to in here. Dunk. Down here, which I think that still works. If it doesn't, you can find plenty of videos online for elongation. But getting to eukaryotes, now it gets a little bit more complex, right? And so if you remember from what we're talking about in terms of transcription and RNA processing, right, we now have a eukaryotic mRNA, which again has a start codon and stop codon, just like prokaryotic mRNAs do. But now we have these 5' UTRs and 3' UTRs and the poly A tail, right? And so these are all important. Oh, and a 5' cap as well for the initiation of translation. Because first we have basically the small subunit assembles on the 5' cap with the help of cap binding proteins, right? These are proteins that directly recognize that 7 mpg or 5' mpg cap. And also, and this is kind of weird in a way, the 3' UTR is also important in translation because this mRNA actually bends round and these proteins kind of assemble to help kind of stabilize this process, this whole kind of complex. And so in eukaryotes, the ribosome does not start on the start codon. It starts at the 5' end of the 5' UTR. And it's going to process along that mRNA until it finds the start codon. So that's one of the big differences between eukaryotes and prokaryotes in terms of initiation of translation. So that's a general overview. What does it look like in specifics? And I think probably I'm going to call it a day after going through this because I think you're probably all a little bit blasted by science and there's no point kind of trying to cram too much stuff into your brains because it's not going to stick. So essentially in eukaryotes, it's actually kind of neat. It's almost like a barcode scanner in a sense. And so we have a whole bunch of extra initiation factors. These are all called EIF something. And so these bind to the cap. And what they're going to do is they're going to recruit the small subunit to the mRNA. So this is kind of analogous in a way to IF3 up here, right? Kind of, right? So this helps recognize the Shine-Delgado sequence and stop the large subunit binding. In eukaryotes, we have a whole bunch of other proteins, cap binding proteins essentially, which will recruit the small subunit of the ribosome. Right? And this is small, but it's not a small protein. This is actually kind of huge and also actually has ribosomal RNA present in it too. And so all of this jazz, including our tRNA for methionine, right, which there's our anticodon again. Here's our amino acid. This is called the initiated tRNA, which kind of makes it sound fancy, but it's really just the first one. All of that assembles to make this initiation complex, right? So we've got a bunch of other initiation factors, right? So not the cap binding proteins. These are just responsible for recruiting all of this jazz. But these other initiation factors, and essentially that whole thing is going to process along the mRNA. And really what you're using is the anticodon of this tRNA to scan the mRNA. And so as you would expect from that, there are no other AUGs in the 5' UTR. The very first AUG or start codon is the start of the open reading frame for that protein. And this start codon is very important, not only because that denotes where the protein will start to be synthesized, but it also sets the frame for the rest of the protein. And so if you kind of like squint in and look at it, this is just a collection of letters. AUG, UUU, GAA, GUG, whatever, right? There's nothing intrinsically, apart from that start codon, which will tell the ribosome, hey, this is going to be UUU is a word, right? It could be UUG is a word, or even UGA. Well, shit, that's a stop codon. We don't want that, right? And so the only thing that tells the ribosome, hey, the next word is UUU, and the next word is GAA, the next word is GUG, is that start codon. Because from that start codon, the ribosome will read the mRNA in groups of 3 letters, 1 codon at a time. So this is super, super important, because otherwise it's just a collection of letters. And that's also why, oops, let's go back up. If you were to add or remove a letter through an insertion or deletion, you change the frame. And that almost always screws up the protein. Well, no, actually, it always screws up the protein. Because if you imagine here, we've got AUG, UUA, UGA. So if we were to take out 2 U's or add another letter in between, the next codon after that would be UGA, which is a stop codon. Translation stops, no protein, right? So those kinds of mutations are really, really deleterious, because this frame is really in reference to only the first codon, which is AUG. Those are called frame shift mutations. Anyway, so the whole caboodle kind of trundles along quite happily, chunk, 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 until it finds a AUG, right, which you can see here. When it does, everything else assembles, right? We have our large subunit coming in, right? And so we've got a whole bunch of ribosomal RNAs in here, as well as a whole ton of other proteins. We have all of these initiation factors release, including from the cap. And now we have our tRNA for methionine in the P site. This is the same as for prokaryotes too. Just the way in which you get there is different. As soon as that occurs, now the A site, the access site, is open. Oh, it's phenylalanine. There you go. That's the next one. And the next tRNA will enter that matches that codon, that UUU, which happens to be the tRNA for phenylalanine. Once it's there, peptide bond is formed between methionine and phenylalanine. The amino acid is cleaved from the initial tRNA. The whole thing moves over. 3 bases. Next codon will be GAA, whatever that encodes. I can't remember these from memory. Then the same will happen. Now we're going to have a dipeptide with methionine and phenylalanine. That will form a peptide bond between phenylalanine and the next amino acid. The amino acid will be cleaved from this tRNA, which is now in the P site. move along one. This tRNA then leaves, another one comes in, over and over and over and over again. And as soon as this cap is cleared, or in bacteria, as soon as the start codon is cleared by the ribosome, another ribosome can assemble and start translating that mRNA. So just as you can have multiple RNA polymerases transcribing a gene, you can have multiple ribosomes translating an mRNA. Okay, I think that's enough for today. I'll give everybody that has OChem exam in 15 minutes time to kind of splash some water on their face, inject some caffeine, and scan their notes, I guess. So for next class, we're going to be going through essentially the rest of this, which isn't going to take super long. There you go. That's what that looks like, polysome. And we'll finish this off, and there should be time at the end for questions. So bring questions with you, and I'll be able to answer them and kind of use that essentially as another study session for your studying for exam 3. Exam 3 will be very heavy on all of this kind of nuts and bolts stuff, right? And so there's a lot to put together and a lot of potential confusion between different things going on, which you need to make sure are clear in your head. So bring that with you on Tuesday, and we'll spend as much time as we can going through those questions. All right. Have fun, everybody. Good luck in your exams for everybody in OChem, and see you next week. Have a great weekend.